Enzymes have 6 classifications, namely oxidoreductases, transferases, hydrolases, lyases, ligases, and isomerases. For this presentation, transferases will be discussed. By definition, it catalyzes the transfer of a functional group from one molecule to another. Transferases helps the donor molecule transfer to an acceptor molecule. It is grouped into an easy number of classifications depending on the chemical reactions they catalyze. For example, methyl transferases is under EC 2.1, which are enzymes that transfers methyl groups. Transferases is an integral part of the life processes in our biology. Hundreds of different biochemical pathways involve transferase and so. Deficiency on this type of enzyme results into various diseases and complications. Generally, in a transferase, the X group is the donor and Y is the acceptor. These groups are the functional group that needs transferase activity. The photo below shows NH3 plus group on your left, a transferase enzyme in the middle, and the alanine reactant in the right. The NH3 plus group is the donor group, while the alpha-ketoglutarate reactant is the acceptor group. Through the alanine transaminase, the ketone functional group represented by the red-lettered symbols is transferred to the other molecule. As stated in the definition, transferases is involved in several biochemical processes in the body. Specifically, transferases catalyzes the transfer of dysfunctional groups: methyl, hydroxymethyl, formal, glycosyl, acyl, alkyl, phosphate, and sulfate groups. Aside from that, transferases have a vital role during translation of proteins. The peptidyl transferase catalyzes the two principal chemical reactions of protein synthesis: peptide bond formation and peptide release. The 6 types of enzymes, along with the biomolecules, coexist with one another inside the human body. Likewise, different types work with one another to achieve their end goal. As an example, let us explore the cellular respiration, which involves several metabolic reactions to convert energy and release waste materials. It has 3 principal stages that utilizes transferases, oxidoreductases, lyases, and hydrolyses. The 3 stages are as follows: glycolysis, Krebs cycle, and oxidative phosphorylation. In glycolysis, transferases such as hexokinase, phosphofructokinase, and pyruvate kinase are used as to break down glucose much faster. After that, all end products move to the aerobic process called Krebs cycle, which makes use of oxidoreductase enzymes like succinate to catalyze oxidation to fumarate. For the last stage, several oxidoreductases are involved in the process. The diagram shows the different enzymes on the cellular respiration along with the specific name of the enzymes. There are a lot of diseases and defects that people can get if they do not have enough transferase enzymes. One of that is the metabolic disease called succinyl COA3, keto acid, CoA transferase deficiency, or simply SCOT disease. People with SCOT disease experiences changes in the OXCT1 gene, making them unable to produce the SCOT enzyme. As this happens, the body can't break down ketones, the substances produced in the liver when fats are broken down. As a result, the body does not produce enough source of energy and the ketones begin to accumulate in the blood.